Mr. Wilson here, Anatomy, Chapter 9. This is Part 2. We're starting out here talking about the synapse. The synapse is the connection, it's the system of the cell that's sending the message. So it's the end of the first cell and the space and the beginning of the next cell. That whole system is called the synapse. The cell that is at the beginning of this is called the presynaptic cell because it comes before the synapse. And then the other one's called the postsynaptic cell. And then the actual space between there is called the synaptic cleft. At the synapse, there are neurotransmitters inside of the presynaptic cell. They're kept inside of synaptic vesicles, which are like little sacs. And then they bind to the cell membrane of that presynaptic cell and release their neurotransmitters out into the synapse. And the action of neurotransmitters, there's two main classes of neurotransmitters, these little chemical signals. They're either excitatory, which means they're driving the membrane potential toward firing an action potential, an impulse, or they're inhibitory, which means they're driving it away from an impulse. So you can see in the illustration here, here's our presynaptic cell, axon terminal. This is the synaptic knob. Inside of there, there's lots of mitochondria. Remember, those are the little powerhouses of the cell. And then there are those synaptic vesicles. Those contain the neurotransmitters. They bind to the cell membrane and then spill their contents into the cleft. They travel across that space and they bind to receptors on the postsynaptic cell. Membrane potential. So the cell membrane in a neuron is typically polarized, which means it's not got an even distribution of charge. It's got sort of a positive end and a negative end, and it sits that way at rest when it's not sending an impulse. The analogy to kind of remember this is a salty banana. I don't know who came up with that, but I've been using that for years to kind of remember where the ions are. So salt we know has sodium, and bananas we know have potassium. And if you had a salty banana, you'd have it because you'd put salt on the outside, and the potassium in the banana would be on the inside. And that's the way the membrane potential is for the cell. There's a higher concentration of sodium on the outside and potassium on the inside. We've got different cell membrane channels that work to maintain this. So the resting potential, which means that the cell's not sending an impulse, is negative 70 millivolts. So the sodium potassium pump works when the cell is at rest to maintain that imbalance, to maintain that negative 70 millivolts. Because if diffusion just took over, it would come to balance, it'd come to equilibrium, there'd be no potential. So the sodium potassium pump is working to do that. It takes ATP to maintain that resting potential at negative 70 millivolts. When stimulation occurs, the membrane becomes depolarized. So again, at rest, it's polarized at that negative 70. As it's becoming depolarized by the action of those neurotransmitters that are excitatory, it drives that potential up in a positive direction from negative 70 to negative 55, which is the threshold potential. Once it hits negative 55 millivolts, then it's going to fire an action potential. You can see here this is a local potential, which means this is just happening at a spot on the axon and it doesn't get enough stimulation to fire the potential. So there it is at rest, negative 70. It gets excited toward threshold, but not all the way to threshold. If it gets all the way to threshold, we'll see a big action potential on that graph. So when it gets to threshold and it fires that big action potential, it becomes depolarized, and it actually goes up beyond zero, even up into the positive. Then after the depolarization, there's a period of repolarization where the slope becomes negative again and it's driving back down toward resting potential. And then quite often it goes beyond that. It goes even lower than negative 70 for a little while. That's called hyperpolarization. and then it balances back out and comes to resting potential there at negative 70 millivolts again. So this sequence of events is called an action potential. The depolarization, repolarization, hyperpolarization, and you can see that here on the graph. So it's at rest negative 70. The action of the neurotransmitters is driving it toward that negative 55. Once it gets there, then it gets this large depolarization, which is the positive slope portion. All the way up here you see to about a positive 30. And then the repolarization, the negative slope, bringing it back down toward resting. There's that small period of hyperpolarization where it's actually below negative 70, and then it evens out back to normal. I've got another illustration there to show that. And yeah, so that will just continue on down the axon until it gets to the end. All right, guys, go Knights.